Hello Bio 201 and welcome to a special YouTube version of your lecture on Chapter 5, Membrane Transport. So as you have realized by this point, we're not going to be meeting tomorrow, Monday, October the 14th, actually probably the day that you're actually taking a look at this video, hopefully. But instead we're going to cover the material that we would have covered in this particular format so we don't get behind. So as you recall, just before this past exam, we left off with this particular slide. We talked about the general characteristics of plasma membranes, and I don't believe we'll go back and review that at this particular time, but at this point you're extremely familiar with plasma membranes and the components of plasma membranes, including phospholipids, proteins, and the cholesterol that we mentioned briefly before we closed on Wednesday, I guess before our last exam. What we really want to start with today is a short discussion of these membrane proteins. And so you're aware of the fact that there are proteins that span the plasma membrane in many cases. Or in other cases, they're just embedded in one portion of the membrane and don't go from one side to the other. And so the question is, what do these proteins do? And the answer is long and complicated. But we can narrow it down to just a couple different functions that we want to talk about as we begin our discussion of transport across the plasma membrane. So first off, we have channel proteins. We'll talk about channel proteins over the next few slides. Channel proteins allow molecules to go from one side of the membrane to the other. In most cases, but not every case, this is a protein or a molecule rather, that's moving across the membrane that would not otherwise be able to move across the membrane. And so the channel protein makes it possible to go across the plasma membrane. A carrier protein is a type of channel protein, and so a carrier protein binds to a molecule that needs to be transported across the membrane and allows it to make its way across that particular membrane. You may say, well, what's the difference between a channel protein and a carrier protein? A channel protein allows molecules to flow through, whereas a carrier protein typically binds to one molecule at a time and transports that molecule across. Hopefully that makes some sense. So for instance, channel proteins function in your nervous system. I've mentioned to you before, and you may remember our discussions about the nervous system. And so you may remember that a nerve impulse is actually the movement of ions back and forth across the cells of your nervous system, and they are able to move as a result of these channel proteins. Sodium and potassium ion channels open up, and a tremendous number of ions, thousands of ions, flow back and forth, even through one channel across that membrane. And so that's different than a carrier protein. A carrier protein may be a protein such as one that delivers glucose into the cell. And so a carrier protein binds to one glucose molecule and moves that particular molecule across into the cell one at a time. And so they are similar but somewhat different. And next we have cell recognition proteins. We've talked about these pretty thoroughly we did talk about these pretty thoroughly on Wednesday. And so glycoproteins, you're aware of the fact that glycoproteins serve as name tags in allowing the immune system of the body to distinguish self from non-self. We talked about the interesting experiment in which you can do a transplant on a neonatal mouse before it's born, and you can transport the tissue from another mouse into the mouse of that body, and that neonatal mouse will then recognize that tissue as belonging to it because of the fact that it now recognizes the glycoproteins on that foreign mouse. But your immune system cells, as I mentioned Wednesday, types of white blood cells, are wandering through your body right now looking at your cells, and they rely on the glycoproteins to determine what cells are supposed to be there and what cells aren't. When they find cells that aren't supposed to be there, they of course destroy them through a number of different mechanisms that we really don't talk much about in this class, but that you'll learn about later. Receptor proteins are so important in biology. So much of physiology is simply a discussion of these receptor proteins. Receptor proteins bind with specific molecules, and so you may perhaps we haven't discussed this in this class, but when you think about receptor proteins, think about a lock and key sort of interaction. The key would represent what we call the ligand, L-I-G-A-N-D. The ligand binds to a receptor, and activates that receptor, bringing about some response in a cell. And so hormones universally work on, using this concept of receptor proteins. Hormones move through the body, bind to a protein, a receptor protein, and bring about some change in the cell as a result of that binding. You know, it's perhaps useful to think back to our discussion of estrogen mimics and endocrine disrupting compounds in our pollution chapter at this particular point. Remember how I told you that you should not be drinking water out of those water bottles that have set, say, in your car in the sun, because those estrogen-like plastic molecules leach out into the water, and they look like estrogen, and so they bind to the estrogen receptor. That's not supposed to happen, but some receptors are not as specific as others. The estrogen receptor tends to bind to the ligand that it's supposed to bind to, which is estrogen, and other ligands as well. Enzymatic proteins, and so we actually have enzymes here. Enzymes are going to be an exciting discussion that we'll get to very soon in our class. And so many enzymes are actually embedded in the plasma membrane. And so in this case, these are proteins that are not spanning the membrane most likely, but that are working on the inside or outside of the actual membrane. I'm not going to describe enzymes too much at this time, and so don't worry about that right now, but we'll come back to that in a future chapter very shortly. Finally, we have junction proteins. Junction proteins in many cases span the plasma membrane and attach two adjacent plasma membranes to each other. And so these are specialized proteins that are not doing anything in terms of actually moving, but rather they're simply joining two adjacent cells together. And so these are some interesting examples of the functions of these membrane proteins. Remember, these are going to be extremely important as you move ahead in the field of biology and learn more and more about how things like cells and the human body work. So here we have some examples. Perhaps I should have gone to these as we had our discussion. I'm going to use the cursor here to point a little bit. Here we have a channel protein. A channel protein is forming a channel allowing molecules to pass through. Here's a carrier protein, which is grabbing molecules one at a time and moving them through the plasma membrane. Here's your cell recognition protein. The major histocompatibility complex, MHC, glycoproteins, are the primary name tags of the cell. We haven't mentioned those MHC complex proteins, or that's redundant actually. The MHC stands for major histocompatibility complex. We haven't mentioned those yet, but those are the primary cell recognition proteins of the body. Here's a receptor, nicely drawn. This is the receptor, and then this is the ligand to which I referred. Here's an enzyme, and so this enzyme actually does span the membrane, but its function has nothing to do with the fact that it spans the membrane. So here we have a substrate binding to the active site of the enzyme, and the enzyme is turning that substrate into a product. And so that's a nice example of an enzyme protein. And here are some proteins that form tight junctions in the between cells. And so tight junctions are found in areas such as your stomach, the lining of your stomach, where it's essential to keep hydrochloric acid and the powerful enzymes in your stomach inside the lumen, inside the open area within the stomach. And so those are proteins that form these junctions that do hold the adjacent plasma membranes of cells together very tightly. Alright, so now we get into an exciting discussion, which is the permeability of the plasma membrane. And so you know a little bit about this at this particular point. You are aware of the fact that some things can move across the plasma membrane, as we see here in this particular bullet, and other things cannot move across the plasma membrane. And so the question becomes, what can and cannot move across the plasma membrane? Well, if I was with you right now, I would ask you, what is the region that I'm pointing to with the cursor right now like? Is it water-loving? Is it a water-loving or water-hating region? And you would say this is a water-hating region, because you know that the hydrophilic water-loving heads of the phospholipids point outward towards the water outside the cell, and the inner layer point inward towards the cytosol, which is mostly water inside the cell. So, in summary, out here where the cursor is now and down here where the cursor is now, those are water-loving layers, and here inside the plasma membrane is a water-hating region. So, let's go over what molecules can and cannot pass across the plasma membrane. First off, if you're big, if you're a macromolecule such as a protein, a carbohydrate, you're not going to be able to go across that membrane. You're just too big. The molecules will not part to let you through. The phospholipids won't part. And so you see that the macromolecule cannot pass through the membrane. On the other hand, let's go over here. If you're charged, if you're an ion, that makes you soluble in water, and that means you cannot pass the plasma membrane. So ions cannot make it through this lipid-loving and water-hating layer, this nonpolar region that I'm pointing to here. So large, if you are large and if you have a charge, then you are not able to pass through the plasma membrane. All right, now on to what can pass the membrane. If you don't have a charge, as this purple molecule here fits into that category, you can pass the membrane. You can go through the membrane. And so this means that you are lipophilic or hydrophobic. You dissolve just fine in lipids, and so you can pass through the membrane without any trouble. But you've got to be small, remember? So if you're small and non-charged, then you can pass through the membrane. Or, interestingly, if you are just really small and are somewhat polar, then you can pass through the membrane. And so water can, to some extent, pass through the membrane. We're going to see later there are special proteins that insert into the plasma membrane to increase water permeability, but water can, to some extent, actually pass through the membrane, which is actually essential to the discussion that we're going to have in just a few minutes. Water molecules can pass through the plasma membrane. All right, back to our bullets over here. If we think about the ways that molecules can pass through the plasma membrane, we divide them up into passive and active transport. Passive transport does not require energy. Molecules simply follow a concentration gradient, whereas active transport does require energy. It takes energy and it takes a carrier protein. In the case of active transport, you're pumping molecules up a concentration gradient, whereas in the case of passive transport, molecules are flowing down a concentration gradient, as we'll see illustrated more clearly on the future slides here. All right, so remember we have two broad categories, passive and active transport. Passive transport then describes both diffusion and facilitated transport. You can see over here on the left-hand side it says energy not required. And I'm going to use the term up and down a concentration gradient quite a bit over the next few slides here. And so you realize that down a concentration gradient implies that you're going from an area of high concentration to lower concentration. That's not surprising. And so things like lipid-soluble molecules, the gases, can diffuse across the cell. And so oxygen, for instance, is reaching every cell, every living cell in your body at this particular moment as a result of diffusion. Facilitated transport we'll go over a little bit more detail in just a moment. It's still passive transport, but it's a molecule like a sugar or an amino acid that can't make its way across the plasma membrane on its own. So it does require a carrier protein to get it across the membrane. Again, we'll come back to that in just a moment. Now we have two types of transport that require energy. Active transport, which we see here, is moving up a concentration gradient from an area of low concentration to high concentration. So sugars, amino acids, and ions move up a concentration gradient via active transport. And then bulk transport. Bulk transport moves a lot of molecules across the cell at one time. And so we'll talk about phagocytosis, for instance, in just actually a little bit later in our lecture. We'll talk about phagocytosis, which is a type of bulk transport. All right, what about diffusion? And so what we see in the case of diffusion is this, that we have to have two things, a solute and a solvent. A solvent is a liquid, and a solute is a very small molecule that's dissolved in that liquid. And so diffusion moves molecules from an area of high concentration to an area of lower concentration. And so we're moving down a concentration gradient. In the case of diffusion, we're always moving down a concentration gradient. Here's a nice little example from our text. We take some goldfish. Not sure exactly what the function, the purpose of the goldfish in this example is, but they're mildly amusing to note there. So if we take a goldfish bowl and we drop some dye that's hopefully non-toxic in there, then that dye is going to spread out via diffusion until the whole goldfish bowl is a light green color. And so you can see what's happening here. The molecules are packed tightly together. They start to spread out, and then they eventually achieve an even distribution in the goldfish bowl there. And so keep in mind that the mechanism that allows diffusion to occur is simply the random movement of molecules. Molecules are constantly in motion, and they're going to move until they run into something and bounce off. And so when you drop that dye in there, the molecules are tightly packed together, but they're going to move until they hit something. And so eventually that random movement means that they spread out and color the entire goldfish bowl green. So that's what we see regarding diffusion. I believe that we have covered everything there. Molecules are moving both ways along the gradient, but the net movement is from high concentration of molecules to a low concentration of molecules. When equilibrium is eventually reached, then net movement stops. The molecules are still moving, but they've achieved an even distribution, so they're no longer going from a high concentration to a lower concentration. All right, and so this is keeping you alive at this moment. You'll be interested to realize that the whole purpose of breathing is to bring oxygen, or rather air, that has a relatively high 21% concentration of oxygen into your lungs. That oxygen diffuses into your blood via the capillaries, and so this is an alveoli, an alveolus singular, alveoli plural. And interestingly, these are tiny little microscopic air sacs, and so air goes all the way down into these, and these are actually very sensitive, and that's why working in a coal mine or smoking or breathing asbestos is very harmful for your body, because these little particles get clogged down here, and eventually, although you have millions of these in each lung, eventually you lose those and lose lung capacity the more that you're exposed to. But they function by, here's the hole that air comes in, oxygen comes into the alveolus, and then oxygen goes from a high concentration in the alveolus to a lower concentration in the blood. And then that oxygen goes back to your heart and all over your body, and that's how your body's functioning at this particular point. Diffusion is moving oxygen from the blood from the air into your blood, and then once it gets to the cells of your body, oxygen again by diffusion is going from the blood into the cells of your body. It's also interesting to note that, right here we have oxygen going from a high concentration in air to a lower concentration in blood, which is a liquid. And so diffusion can occur across different mediums. Okay, if I was with you, I would ask if there are any questions at this point, but since I'm not, let's go ahead and go on. Osmosis. Osmosis is a very important topic, and one that we'll go over here for just a few moments. Osmosis is going to be a fundamental concept that you absolutely have to understand in order to understand how any type of biological process, whether it's an organism living in the environment or whether it's how the human body functions. Osmosis underlies much of the field of biology. So osmosis is a special case of diffusion, oftentimes described as the diffusion of water, which is an accurate description. Osmosis occurs, as we see down here in the third bullet, osmosis requires a couple things in order for it to occur. One is a selectively permeable membrane. A membrane has to be present that allows some things to pass through and other things to not pass through. Now, in practical terms, what we're always talking about in the field of biology when we talk about osmosis is a plasma membrane, and we are talking about the movement. Water can move across the plasma membrane, but solute cannot move across, such as salt cannot move across the plasma membrane. But they're all different types of solutes. And so in order for osmosis to occur, there has to be a solute concentration difference on each side of the membrane. Let's take a look at this diagram and attempt to explain osmosis. This is a point where I like to draw on the board, but we can do some of that on Wednesday. But let's take a shot at getting a good understanding at this point of osmosis. So Here we have what we refer to as a thistle osmometer, as you'll be excited to know. A thistle osmometer. So named because it looks sort of like a thistle. And in the thistle osmometer, we have a solution that has 10% solute. Outside the thistle osmometer, we have a solution that has 5% solute. So there's more solute inside the thistle osmometer than outside in the beaker. So the thistle osmometer, as you can see, is submerged in the water, and the glass, of course, of the thistle osmometer is non-permeable to everything. But the bottom area, shown in red, is a differentially permeable membrane, or semi-permeable membrane, as illustrated here. And so the solute can't escape. There's going to be 10% here and 5% outside, at least until water moves. So the solute can't move back and forth, but water can. Water can move back and forth. And so the question is, this is what I'd like you to ask yourself. Where is water more crowded? Is water more crowded inside the thistle osmometer with 10% solute, or outside the thistle osmometer with 5% solute? The conclusion you'll have to come to is that water is more crowded outside. In fact, look over here at this part of the diagram. If you haven't noticed, I'm pointing with the little cursor here. The green particles represent solute particles, and of course the blue particles represent water. And so you'll notice that inside the thistle osmometer there are more green and less blue. So water is less crowded there. You may say, well, that doesn't make sense to me because there's solute, so it seems like there's the same amount of space being occupied. And that is true, but you'll just have to accept the fact that water behaves in this particular manner. Where there are more water molecules, the water molecules want to move to an area where they are less crowded, where there's more solute and less water. And so that means that the water molecules move from the 5% side of the membrane to the 10% side of the membrane. And so what occurs is that the concentration of solute inside the thistle osmometer shrinks. The concentration of solute outside the thistle osmometer goes, did I say that right? Yeah, shrinks, goes up actually, because we're losing water. So it's above 5% now and below 5% inside the thistle osmometer. So water's moving in. That means that we started with 10%, but now we have less than 10% because we've added water but have the same amount of solute. We have more than 5% outside because we've removed water and had the same amount of solute. And so that's a nice illustration of what's occurring here. And let's introduce a couple terms that relate to this process. These are very simple terms: isotonic, hypotonic, and hypertonic. These are terms that refer to solute concentration. Remember, this refers to solute concentration, not to water concentration. Isotonic. In an isotonic solution, we have the same solute on both sides of the membrane. Hypotonic. In a hypotonic solution, we have less solute on the hypotonic side and more on the other side, which we refer to as the hypertonic side. And so interestingly, if we place a cell into a hypotonic solution, that means that there's more solute inside the cell and less solute outside the cell. Remember, listen to my wording. We placed a cell in a hypotonic solution. And so you can ask yourself, where is water more crowded? Well, water's more crowded where in the hypotonic solution, outside the cell, so water moves in. And that means, as you can see here in this third bullet, the cell is going to swell and perhaps break. And we call that lysis. If it does break, then the cell would be lysing. It would undergo lysis and rupture as a result of the water moving into this cell, the solution, solvent, which is water moving into the cell. Okay, and so that's the difference between hypertonic and hypotonic. Oh yeah, and then hypertonic, if we place a cell in a hypertonic solution, then the cell will shrink. And it's, if you think this through, I think you'll agree, it has to be the opposite of what occurred in the hypotonic solution. So this means that the cell will contract. And something may occur called plasmolysis. Plasmolysis is a special situation that occurs whenever a plasma membrane pulls away from a cell wall. This occurs when you take a cell that has a cell wall, like a plant cell or like a bacterial cell, and place it into a hypertonic solution. The cell wall is permeable, so water can move through that cell wall, and then the plasma membrane is semi-permeable. The solute and cannot pass through the plasma membrane, as you know. There's no difference in the plasma membrane here as opposed to what we talked about previously. So this means that the water moves through the plasma membrane, solute doesn't, and the plasma membrane shrinks away from the cell wall, creating something that we call plasmolysis in a cell, remember, that has a cell wall. This occurs in bacterial cells, and this occurs in plant cells. Alright, so here we see these processes illustrated quite nicely. In an isotonic solution, there's movement in and out of the animal cell that we see on top here. So there's no net movement. The animal cell doesn't shrink or burst. In a hypotonic solution, water moves in and the cell bursts. In a hypertonic solution, water moves out and the cell actually shrinks. Here we see a plant cell. A plant cell in an isotonic solution, in this case there's no net movement of water. Water moves in, water moves out, nothing exciting happens. In a hypotonic solution, water moves in, creating this thing we call turgor pressure. Plants actually do best in a slightly hypotonic solution. So water moves in and the central vacuole pushes out against the cell wall, creating this thing again we call turgor pressure. And here's plasmolysis that we just talked about. Put a plant cell in a hypertonic solution and plasmolysis occurs. Alright, and so we're going to wrap up our discussion today with a brief discussion of facilitated transport and then active transport. We're done with our discussion of osmosis. Facilitated transport takes place when you have a molecule that cannot make it through a plasma membrane on its own. And so in this case you need a carrier protein to get it across the membrane. This occurs with glucose, for instance, in many cells of the body. Glucose can't pass across the membrane, but it does. A concentration gradient does exist. There's more glucose outside the cell than inside, so it does want to flow into the cell. But again, it can't because it can't move through the plasma membrane. So here's where our carrier protein enters. A carrier protein binds to glucose and allows it to flow through the cell membrane. Does facilitated transport require energy? The answer to that question is no, it does not. This is because in the case of facilitated transport, molecules are still moving down a concentration gradient from an area of high concentration to an area of low concentration. It's just that they can't pass through the plasma membrane on their own, so they've got to have that carrier protein. Active transport is completely different. In active transport, we have to have a carrier protein, but we're moving molecules against a concentration gradient from an area of low concentration to an area of high concentration. And this does require energy. We're pumping molecules. Think about if you went below a dam and you tried to pump water back up above that dam. Water will flow over the top of the dam, right? Quite easily to the bottom of the dam. If you think about a big reservoir like Lake Cumberland, for instance, water will go from above the dam to below without requiring energy. But if you want to get water from that river down below the dam back up into the lake, which is actually a reservoir, as you know, then you've got to have some energy and a pump to get it back up there. So active transport does require energy. Active transport is extremely important in many different processes in your body, but I think the nervous system, again, is a great illustration of this. Active transport is what keeps your nervous system functioning. Every time a nerve impulse moves through your body, active transport restores that ion gradient within a few fractions of a second so that you can undergo another, what is called an action potential, another movement of ions. And in other words, another impulse can pass within just a few seconds. Let's take a look at this picture, and this will be the end of our lecture today. So here we have a carrier protein, and what process, ask yourself, what process is being illustrated here? And you'll come to the conclusion that this is facilitated transport. Very good. And so facilitated transport occurs when a molecule goes from an area of high concentration, that's how you could tell it was facilitated and not active transport, to low concentration. There's 4 of the molecules over here, none over there. Here's our carrier protein. So the molecule binds to the carrier protein and then is expelled on the other side of the membrane. Interestingly, carrier proteins are typically very, very specific. They typically carry only one type of molecule across the membrane. And that's it. Just one type of molecule. All right, and so what we'll do on Wednesday, is we will finish this particular chapter. We won't have a quiz on Wednesday, but please remember that your Biology Through the Eyes of Faith questions are due on Wednesday, or is it Thursday? Sorry, I can't check from this particular point, but it's either Wednesday or Thursday. They're due before fall break. I believe it's actually Thursday. And so make sure that you finish those and get them turned in. Remember, that's a big part of your grade, relatively speaking, and a very easy part of your grade. You definitely don't want to miss those points. So I will see you again on Wednesday.